This is Jessica and Nick. This week we're going to be presenting on week 7 of spatial ecology lecture and reading materials. This week surrounded scale and selection.
During this week we learned about habitat quality. An example we have is we're going to use just basically numbers, which is habitat 1. It has the cues 1, 2, and 3, which we will consider higher quality cues, while habitat 2 has 4, 5, and 6, and not 1 and 2. In this case, habitat 1 would be considered high quality, thus the species would show a strong preference for it, while habitat 2 doesn't hold the cues, and even though the habitat is suitable, it would not be the chosen area.
A real-world example is pictured here on the right. This is the border between the island nations of the Dominican Republic and Haiti. Obviously, you can see that the Dominican Republic, pictured on the right, would have more habitat cues than its Haitian counterpart. Because of its higher agrarian habitat and highest forest preservation, there is more biodiversity present, and species are more likely to choose this side of the border when given the opportunity.
We also learned about how these cues are integrated. We learned about summation, which is by Hiddlin, and each cue is added or subtracted to form a total score for a habitat. If the score exceeds a particular threshold, the animal settles. The second one was niche Giesalt, which was by James. The habitat is responded to as a whole. And then we also have hierarchical selection, which is by Weens and Rottenberry, and it looks at the large scale versus the fine scale of selection. We also learned about natural order. The first order was to select a physical or geographical range of a species. The second order was home range placement of an individual or a group within the species range. The third order is the use of habitat elements within the home range. And the fourth order was resources selection from areas within the home range.
Two types of sampling methods we can use on species include systematic and stratified sampling. Systematic divides the landscape into equal units and samples randomly from the subdivisions, such as the equally sized polka dots in the background on the left side. Stratified sampling takes samples from predefined categories, such as the black and white rectangles on the right side. One important spatial ecologist was known as Hutchinson. His work on scale was monumentous, especially his development of the classical niche theory, which says that resource use could reveal a species niche. One of his PhD students, Robert MacArthur, investigated warblers using different parts of a tree. He revealed that warblers use different parts in a method called resource partitioning, so that several species could use the same habitat. Scale is important in this because of scarce resources and competitive exclusion principles. The island biogeography model was developed by student Robert MacArthur and Edward Wilson. They found that species diversity could be a function of distance and habitat size. Islands or habitats closer to high concentrations of animals and larger in size will have more species richness. This was illustrated using the idea of birds or other migratory animals colonizing nearer and larger islands as opposed to those that were farther away or smaller. Robert MacArthur's work was very monumental. An award was made in his name and once was awarded to Dr. Simon A. Levin. He describes how habitat scale is very important in spatial ecology and could possibly be distorted. His work described this as every organism being an observer of their environment. And this could be problematic since everyone and everything has something called observer bias. He used several examples from across the globe, including plant success, krill in the Scotia Sea, and zooplankton abundance, to illustrate how easy it is to distort scale using different types of observer bias.